Let's check out the construction going on for the new roller coaster ride at Epnot as we cover DNA replication, a topic test riders love. But before we start building stuff, we'll quickly mention a few key things. First, remember that DNA replication is a semi-conservative process. This means that each parent strand serves as a template to make complementary daughter strands. Next, remember that DNA replication specifically takes place in the cytosol of prokaryotes because they don't have a nucleus to store DNA. In contrast, eukaryotes replicate DNA in the nucleus. And last but not least, we need magnesium to replicate DNA. It's a cofactor for some of the enzymes involved. With that said, let's dive in. Prokaryotic DNA replication begins at the origin of replication. First, the pre-priming complex, which consists of helicase and associated proteins, binds to AT repetitive segments at the promoter and origin before partially separating the DNA strands. Next, helicase unwinds the double helix, creating a replication fork. To show that, we've added this helical coil train unwinding the tracks. Note the 3p batteries because helicase requires ATP. As helicase unwinds the DNA, single-stranded binding proteins bind to single-stranded DNA to prevent it from annealing to the other strand or getting broken down by endonucleases. We've added these strong folks holding the track to symbolize these proteins. All right, so we've unwound the DNA. Now what? Well, there's a problem. The unwinding process has created positive supercoils in front of the replication fork and negative supercoils behind it. They're kind of like the tangles you get in your phone's power cord when you're unwinding it. Okay, maybe not, but they're definitely problematic. Fortunately, we have DNA topoisomerases to get rid of them. Let this top hat worker using a single saw to cut the track remind you of type 1 topoisomerase. This enzyme cuts a single DNA strand to allow for passage of another strand through the break in the order to relieve any supercoils before rejoining them. Meanwhile, our other guy here using two saws to cut both tracks is type 2 topoisomerase. This enzyme cuts both strands simultaneously and allows passage of DNA through the break before rejoining them. Unlike Topo 1, Topo 2 requires ATP, just like those battery-powered gyrating saws. Why gyrating? To remind you of gyrase, a specific kind of type 2 topoisomerase found in bacteria. One more thing. You should know that fluoroquinolone antibiotics block bacterial DNA replication by specifically inhibiting DNA gyrase and topoisomerase 4. And gazoon tight to you, sir. Now that we've unwound the DNA and eliminated the supercoils, we need to add a little bit of primer. A base coat, if you will, before we paint our masterpiece. Oh, not that kind of primer. Sorry, RNA primer. Short RNA primers are placed by primase. All right. Enough monkeying around. Once the primers are placed, DNA polymerase 3 can read the nucleotide sequence on the parent strand in the 3 to 5' direction to add new complementary nucleotides at the 3' end of the daughter strand. DNA polymerase 3 is considered a highly processive enzyme, meaning it can synthesize DNA continuously without dissociating from the template strand. To remind you of DNA polymerase 3, we illustrated this worker in the number 3 train laying down blue tracks. Now, you're probably wondering what makes this enzyme so highly processive. 2 words: sliding clamp. The sliding clamp holds DNA polymerase in place so that it doesn't dissociate from the template strand during polymerization. Okay, so what happens if DNA polymerase 3 adds the wrong base? Fortunately, this enzyme can proofread newly synthesized DNA and remove mismatched bases using the 3 to 5' exonuclease, which snips a nucleotide off the end of a polynucleotide chain. To show that, we'll add this inspector removing the mismatched track and replacing it with some fresh, new blue track. Refreshing. Now that we've started building our DNA strand, we gotta get rid of that RNA primer. This is done by DNA polymerase 1, which has 5 to 3' exonuclease activity. DNA polymerase 1 is represented by the worker in the number 1 train removing the orange track and replacing it with blue track. So both strands of DNA are replicated at the same time, and they're oriented in opposite or anti-parallel directions. But the actual replication only occurs in one direction, 5 to 3'. This leads to the development of a leading strand and a lagging strand in the replicating DNA. The leading strand grows continuously because DNA polymerase 3 is moving in the same direction as the replication fork. On the other hand, the lagging strand keeps having to start new little bits of replication, generating Okazaki fragments in the process. This occurs because DNA polymerase is moving in the opposite direction of the replication fork. All right, now it's time to link the Okazaki fragments. This blacksmith welding the two pieces of track is DNA ligase, which creates new phosphodiester bonds to connect the fragments. Like helicase, DNA ligase requires ATP. When all is said and done, termination proteins bind to specific termination sequence sites to complete DNA replication. Okay, let's change gears and review some important differences between prokaryotic and eukaryotic DNA replication. Unlike prokaryotes, eukaryotic cells have multiple origin of replication sites. Since the genome of most eukaryotes is larger, more complex, and linear, having multiple origin sites means we'll be able to replicate DNA more efficiently. Because of this complexity, it shouldn't be surprising that eukaryotes require more proteins to replicate DNA, but the underlying mechanism remains the same. We've illustrated the major proteins and their respective roles in this table. This table shows at least 5 different kinds of DNA polymerases that have been identified in eukaryotes. Note that they aren't labeled numerically, but by Greek letters. Again, the underlying mechanism of replicating DNA is pretty much the same except the players. There's one more extra step for eukaryotes too. Eukaryotes have a special enzyme called telomerase, a specific type of reverse transcriptase sometimes called RNA-dependent DNA polymerase.
Telomerase adds a non-coding, repetitive sequence of DNA to the ends of chromosomes to protect DNA from getting degraded. To show telomerase, we illustrated this teal-colored junkyard magnet adding stabilizing wheels to the end of the track.
Note that each successive round of DNA replication shortens the telomeres because the lagging strand cannot replicate all the way to the end of the chromosome. So once you run out of telomere, a cell generally can't divide anymore. It's a way of building in a limited lifespan for the cell. Now let's go over some clinical pearls. First, having a shortened telomere length is associated with cellular senescence and aging. This is represented by this old track containing only 2 stabilizing wheels. This is also important for cancer cells. These cells usually have a high telomerase activity, which allows them to divide uncontrollably. To help you remember that, we've added our recurring cancer crab logo to the junkyard magnet. Let's quickly mention Bloom syndrome, a genetic disorder that results in disruption of DNA replication. It's caused by a mutation in the BLM gene affecting helicase. To remember Bloom syndrome, think of this blooming flower face behind the helical coil train. Dang, what a creepy flower. Anyways, before we wrap up, here are some key inhibitors of DNA replication in humans. The first are the topoisomerase inhibitors, which are used in the treatment for certain cancers. The toucan attacking top hat worker 1 will remind you of the tecans, irinotecan and topotecan. They block topo 1. And this foreman grabbing top hat worker 2 by the side will help you remember etoposide and teniposide, which block topo 2. Another way to inhibit DNA replication is by blocking chain elongation using nucleoside analogs that have an augmented monosaccharide portion. Examples include cytarabine, represented by the animatronic saber-toothed tiger, and zidovudine, represented by the robotic Princess Izaldi blocking the track. This ride is starting to get a little weird. I'm getting out of here. After a quick review, that is. DNA replication is a semi-conservative process that begins at the origin of replication. After the pre-priming complex successfully binds to the origin, helicase starts unwinding the double helix, while single-stranded binding proteins bind to single-stranded DNA. At the same time, DNA topoisomerases remove any supercoils created by helicase. Next, primase generates an RNA primer before DNA polymerase III starts adding new nucleotides at the 3' end of each daughter strand, while removing mismatched bases using the 3' to 5' exonuclease. Then, DNA polymerase I removes the RNA primer, and ligase joins the Okazaki fragments together.
Eukaryotes in particular utilize a special enzyme called telomerase, which adds a repetitive, non-coding sequence to the end of the chromosome.
High telomerase activity has been associated with cancer, while having a shortened telomere length might suggest cellular senescence and aging. There are a number of drugs that block DNA replication, including the topoisomerase inhibitors and the chain